We are in the middle of a genetic revolution, and through sequencing the human genome and the genome of many, many animals and other species, we now know how many genes we have. We can identify those. Also through sequencing, we've found many unusual and novel features of our chromosomes, our genomes. For example, species called non-coding RNAs, which actually we think are going to be very interesting. So now the task is, first of all, to work out the function of all these genes, all the proteins they make, all these novel non-coding RNAs. But for us, a particular challenge is this. There are many differences in our genomes, in our chromosomes, from one individual to another. And of course, we inherit those differences from our mum and dad. So the big $64,000 question is, what are the key differences we inherit from our mum and dad that influence the differences between one individual and another in their behaviors, their height, their weight, their physiology, all these sorts of things. Also, most importantly for this institute, what are these genetic differences we inherit that make us more or less susceptible to particular human diseases? For example, I might be particularly susceptible to getting heart disease or diabetes or something. Can we identify whether I am and what those genetic differences are that make me more or less susceptible? Somebody else might be more or less susceptible to getting cancer. Can we identify those? If we can, by the way, we can identify people at most risk and bring them in for early diagnosis. But most importantly, we can understand the biology that underpins, what are the mechanisms that underpin our normal biology and our disease susceptibilities. Then we learn something about how humans work, how we get disease, and ultimately, especially working on this hospital side here, we want to bring that into clinical practice, bring it into diagnosis, bring it into treatment, use the information to follow the progress of disease. That's the big aim we have.
Our research team is interested in understanding the biology of melanoma, which is the deadliest form of skin cancer. The incidence of melanoma is rising in fair skin populations, and in particular, Scotland has had an increasing incidence in melanoma. So the genes that cause melanoma in people are very similar to the genes that cause melanoma in zebrafish. The zebrafish is actually from India. It's from the Ganges River. And we use it for a couple of reasons. One is that many, many labs in the world use zebrafish. So there's a big community, scientific community, and we've developed many tools as a community to allow us to study the genetics of melanoma. Melanoma is from a cell type called a melanocyte, and we can see these melanocytes very clearly at the earliest stages of their development in a little embryonic zebrafish, and we can see those all the way through to adulthood. And then we can watch those melanocytes actually go through the stages of melanoma. So it's visually tells us a lot of information. One of the things that's really great for our lab is that we have our lab in the Cancer Institute, but have our FISH in the MRC Human Genetics Unit. So we work within two of those buildings of the IGMM. But I think probably the best thing is the number of people. So you sort of get this critical mass of scientists.
The Medical Research Council is providing about £60 million over the next 5 years. The majority of that will continue to fund ongoing programs in the Human Genetics Unit. However, a significant proportion, 10 to 20%, combined with money from the University of Edinburgh, will allow us to do exciting new science, to recruit world stars, and this is already happening, to buy new microscopes to allow imaging of cancer cells, and to build a brand new PhD program and help us integrate with the National Health Service.
This production is brought to you by the University of Edinburgh.